Yeah, please start the class, ma'am.
Sure. Good evening, everyone. Myself, Dr. Anchal, and today we are having the class over the topic stomach. So here we have some core feature of Oracle, in which we have live classes, in which you will learn elevate learning, in which there will be interaction one-to-one of the faculty with the student. Then another feature of the Oracle is that through the recording session, you can have the access of our subject anytime, anywhere through the recorded sessions also. And we also provide ultimate comprehensive books with CBME based question answers, MCQ. Uh, then there will be standard plans, which will be of 30,000, in which it includes pre-recorded classes and live classes and doubt clarification also. Then we have the this premium package of 12 months. It will be of 50,000, in which there will be pre-recorded classes, live doubt clarification, one-to-one personalized guidance and notes and quick revision also. For 20% discount, you can use my code, that is Anchal20. Then or in Oracle, I have started the series of GIT, in which we have completed till esophagus, till the stages of swelling. And today we are having the class over stomach. So starting from the introduction. So as we know that stomach is the muscular hollow organ of the GI tract of humans and many other animals. The stomach functions as a vital digestive organ. So in the digestive system, the stomach is involved in the second phase of digestion. Followed chewing, it performs a chemical breakdown by means of enzymes and hydrochloric acid. So about enzyme, we're going to discuss in details also what are those enzymes which are present and what are their function. And another thing, we have HCl in the stomach. So HCl is basically the main function of HCl to kill the bacteria which are present or which are comes in GI tract along with the food. So in humans and many other animals, the stomach is located between esophagus and the small intestine. So as we know, the upper opening of stomach is esophagus and the lower opening is small intestine. Basically, the first part of small intestine, that is duodenum. Then it secretes digestive enzymes, gastric acids to aid in food digestion. Then we have the pyrolic sphincter that control the passage of partially digested food chyme from the stomach into the duodenum, where peristalsis take over to move this through the rest of the intestine. So between stomach and intestine, there is a presence of pyrolic sphincter. Whenever this sphincter relax, that time it will get open, then the chyme will move from stomach into the intestine. Then talking about the functional anatomy of stomach. So basically, the stomach is a hollow organ situated below the diaphragm on the left side in the abdominal cavity. The volume of stomach, empty stomach, here we are talking about. So it is 50 ml. Now, under normal condition, it can expand to accommodate from 1 liter to 1.5 liter of solids and liquids, and the maximum capacity, maximum it can accommodate up to 4 liters. Then we have 4 parts: the cardiac region, fundus, body, or pyrolic region. So as we can see in the diagram also, the upper opening from where the bolus will enter from the esophagus, and the lower opening we have, which will deliver chyme to the intestine. So we have this dome shape like structure that is known as fundus. Then we have here this cardiac region. Then this side at the greater surface area, we have greater curvature, and here we have lesser, and here we have antrum, and this space is also known as pyrolic canal. Now, the very important thing I wanted to discuss with you, that in the this pyrolus region, there are some kind of cells are present. The first is the G cells, which secrete a very important hormone known as gastrin. The second, that is also secretes some soluble mucus. So these are the very important things, and you have to remember. Then in the body part, basically in body and both in fundus, there are some different types of cells are also present. The first is the parietal cells that are also known as oxyntic cells. The second, we have the chief cells. The another name for chief cells is peptic cells. And the third cells are surface epithelium. Now, these parietal cells secretes very two important things. The first is HCl and the second is intrinsic factor. Here, the chief cell secretes a very important enzyme that is important for the digestion of protein also, that is known as pepsinogen, and the surface epithelium secretes mucus. That is basically responsible for providing lubrication and motility to the food. So I hope this whole diagram of the stomach is very well clear to the students. So first we have the cardiac region, is basically the, as we have talked, it is the upper part of the stomach where esophagus open. Then we have the opening is guarded by a sphincter known as cardiac sphincter. Then it only opens towards the stomach, and this portion is known as cardiac end. Second, we have the fundus, that dome-shaped structure that we have seen, which is elevated above the level of esophageal opening. Now we have the body, that is also known as corpus, which is the largest part of stomach, forming about 75% to 80% of the whole stomach. Then it extends from just below the fundus up to the pyrolic region, as we have seen already. Then the pyrolic region has two parts. The upper one is known as antrum and the lower one is known as pyrolic canal. Now, the body of stomach ends in antrum. Antrum is continued as the narrow canal, which is known as pyrolic canal, or it is also known as pyrolic end. Now, pyrolic canal opens into the first part of the intestine known as duodenum. The opening of pyrolic canal is guarded by a sphincter known as Pyrolytic sphincter. And as we have seen, the stomach has two curvature. On the right side, the lesser curvature is present, and on the left side, greater curvature is present, as we have seen in the diagram also. So this is all about the morphology of stomach. Then we have the structure of stomach wall. So as we have seen the cross section of gut also, it contains the outer serous layer, muscular layer, submucous, inner mucus layer. So the outer serous layer is formed of peritoneum. It is the outer covering of stomach. Then the muscular layer is made up of some smooth muscles fiber, in which we have circular, longitudinal and oblique fibers. Then in submucous, we have Mesenter's nerve plexus. Now, what is this Mesenter's plexus? In the last class also we have talked about this plexus is basically for providing the motility to food, right? Now, the inner mucus layer lined by mucus secreting columnar epithelium, the gastric glands are also situated in this layer. Throughout the inner mucus layer, small depressions are present, that is known as gastric pits. Now, the glands of the stomach open into these pits. Inner surface of mucus layer is covered by 2 mm thick mucus. Then another thing we have, the function of the gastric glands is to secrete gastric juice. Now, before going through this gastric juice, we need to discuss some important and main property of the gastric juice. So as we know that the gastric juice, the daily secretion of this gastric juice is approximately about 2.5 to 3 liter with plasma, right? 2.5 to 3 liter with plasma, and it's basically the reaction is acidic due to presence of HCl in the stomach. It consists of some electrolytes also, in which it includes sodium, potassium, chloride, bicarbonate, magnesium, phosphate, and many others also. And it contains some enzymes also. Enzymes like chief cells, renin, lipase, gelatinase, urease. Then there are different types of cells that already we have discussed that is present inside the stomach, in which first we have the chief cells. The chief cells, the another name for chief cells is that we have already discussed. The another name for chief cell is peptic cells. So the secretory product of the chief cells or the peptic cells is pepsinogen, renin, lipase, gelatinase, urease. Now these are the secretory products and very important enzyme for the digestion of food. So just for example, the pepsin digest a very important product that is known as protein. So all these enzymes has a very specific role that we're going to discuss in detail. Now the next thing we have parietal cells. The another name for parietal cells is oxyntic. It secretes two important products. The first is HCl. That function already we have discussed. Basically, this is present for providing acidic medium, and also it is responsible for killing the bacteria which is present in the food. Then another thing we have intrinsic factor of Kessel. So basically, this binds with vitamin B12 and help in the absorption of vitamin B12 in terminal ileum of small intestine. Then it secretes mucus cells also, which produce mucin. Then G cells, a very important hormone for digestion, gastrin. Then enterochromaffin cells secretes serotonin, and enterochromaffin-like cells has the very important secretory product that is histamine. So all the function of these hormones also we're going to discuss in the production of HCl also. Now, the function of stomach. So the first and the very important function of the stomach is storage of food. As we know that the main function of stomach is the storage of food. The food is stored in the stomach for a very long period, that is up to 3 to 4 hours, right? And emptied into intestine slowly. As the chyme will produce inside the stomach, then it will get it will get travel inside the intestine. That slow emptying of stomach provide enough time for proper digestion and absorption of food in the small intestine. Then after the enzymes will act in the stomach, then the food will move into the small intestine. There the digestion, basically the food that is present in the stomach and moves to the small intestine, that is basically partially digested, and the final digestion will occur in the small intestine also. And here the absorption of food also occur with the help of structure that is known as villi. Now the formation of chyme. As we know, the peristaltic movement of stomach mix the bolus with gastric juice and convert it into the semi-solid material that is known as chyme. So basically the food that is delivered by the stomach into the intestine, that is known as chyme. You need to remember these things are very important. Okay. Then another thing we have gastric juice act on mainly on protein. Proteolytic enzymes for the gastric juice are pepsin and renin. So basically, what are proteolytic enzymes? So as the name suggests, the enzyme which break down proteins that are known as proteolytic enzyme. So those are pepsin and renin. So as we can see in the chart also, pepsin, gastric lipase, gastric amylase, gelatinase, urease. So the activator of all the enzymes is acidic medium, and this acidic medium is due to the presence of HCl. Now the pepsin will act on proteins to break it into and convert it into protease, peptones, polypeptides. The lipase, as the name suggests, lipase, so means it will act on lipids, triglycerides of butter, and the end product we will receive is fatty acids and glycerol. Then we have amylase. So for amylase, we have a substrate that is starch, and the end product we will receive is dextrine and maltose. Then for gelatinase, the substrate is gelatin, collagen of meat, and the end product we will receive is peptides. And for urease, the substrate will be urea, and the end product we will receive is ammonia. So basically, what is the main motive of the gastric juice and these enzymes? The main motive to convert these products into the simpler and more soluble form so that the absorption can be done. So as we know, the HCl is present. Now, if it is hydrochloric acid, so means it is an acid with a very low pH. So it is harmful, right? It will destroy the lining of intestine. So for the product, for the prevention of the exposure of this HCl, we have a very important lining of mucus. So basically, This is present to protect the stomach wall from irritation or mechanical injury by virtue of high viscosity. It prevents the digestive action of pepsin on the wall of stomach, particularly gastric mucosa. It protects the gastric mucosa from mixture of gastric juice because of its alkaline nature. Now, the another thing we have the hemopoietic function, and again, this is a very important function. So basically what happens, as we have already discussed, this intrinsic factor of Castle, right, which is secreted by the parietal cell, right? So this factor is very important for the absorption of vitamin B12. Now, how this is important? So basically what happened? Whenever the vitamin B12 reaches inside the stomach, this IF or intrinsic factor of Castle gets bind this vitamin B12, and whenever they both will travel in the small intestine, so it helps in the absorption of vitamin B12 in the terminal ileum. So this is how the function of intrinsic factor, or basically this is very important. Now, why this vitamin B12 is very important for our body? Because it helps in the maturation of RBC. Without vitamin B12 and folic acid, RBCs won't mature. So basically, the intrinsic factor of Castle secreted by parietal cells of gastric glands plays an important role in the erythropoiesis also. It is necessary for the absorption of vitamin B12, which is called extrinsic factor, together from the GI tract into the blood. Vitamin B12 is important for the maturation factor during erythropoiesis, and absence of intrinsic factor in gastric juice causes deficiency of vitamin B12, and this condition leads to a particular type of anemia known as pernicious anemia. So basically, in anemia also we have studied two types, which both are associated with deficiency of vitamin B12. The first is megaloblastic and the second is pernicious. So basically in megaloblastic, what happens whenever there is a deficiency of vitamin B12 in diet? So that's why the RBCs does not mature, and that leads to megaloblastic. But in this pernicious anemia, what happened? There is a deficiency of intrinsic factor. So basically the vitamin B12 is present, but due to the deficiency of intrinsic factor, this vitamin B12 won't be able to get absorbed, and that leads to a condition known as pernicious anemia. Then another function of stomach is the excretory function. So basically, the excretory function, like it helps in the excretion of some substances like toxins, alkaloids, matters, which are excreted with the help of gastric juice. Now, the function of gastric juice. So the first function, as we have already seen, the digestive function, various enzymes are present in gastric juice, which help in the digestion of substances, then food. Then we have hemopoietic function, in which we have seen the function of IF. Then protective function of mucus we have seen. Then next we have the function of HCl. So as we have already seen, it kills some of the bacteria entering the stomach along with the food substances. Second thing, that it helps in the activation of pepsinogen. So as we have seen, the pepsinogen is an enzyme which is responsible for digestion of protein. So basically, whenever it enters in the GI tract, it remains always inactive. So for the activation, it requires acidic medium, and that is basically provided by the HCl. So the HCl also responsible for activation of pepsin to convert in pepsinogen to convert it into pepsin. Then, as we have seen that HCl basically provides acidic medium that is necessary for the action of other hormones also. So these are the important function that basically HCl provide in the stomach. Then we have secretion of pepsinogen. So basically, the pepsinogen is synthesized from amino acids in ER, reticulum in sheep cells. Then pepsinogen molecules are packed into zymogen granules, and they are packed by Golgi apparatus. Then when zymogen granule is secreted into stomach from sheep cells, the granule is dissolved and pepsinogen is released into gastric juice. Now, the activation of pepsinogen will be done by HCl. That already we have seen, it will convert it into pepsin. Now, a very important thing and a very important question for the university exam also, that basically how the HCl secretion will take place inside parietal cells. So already we have studied that the HCl is secreted by which type of cells? They are secreted by parietal cells, right? So here we have the interstitial lumen.
Excuse me, ma'am, your screen is not visible, ma'am. You're not sharing your screen.
Okay, wait. Is it visible now?
Yes, ma'am.
Yes, ma'am.
I think then you have missed a lot. You should have informed me before. Fine. So here we are discussing the mechanism of HCl secretion. So as we can see, this is the parietal cell. Then here we have the interstitial fluid. This is which is basically plasma. Then this is the lumen site. Okay. Then here in interstitial fluid, basically the blood vessel here is present. So what will happen with the process of diffusion, the CO2 will diffuse inside the parietal cells and already H2O is present. So together H2O and CO2 will form a very important component, that is carbonic acid, H2CO3, in the presence of an enzyme known as carbonic anhydrase, right? So CO2 and H2O will form a very important product that is known as H2O. Now, what will happen in the presence of this same enzyme, carbonic anhydrase, this product, yes, this product will get split into two components. The one we have bicarbonate ion and the another we have H plus, right? The one we have bicarbonate and the another we have H plus. Now, what will happen? This bicarbonate will get or secreted into the interstitial site and in the exchange of an ion known as chloride, right? So basically this channel is known as bicarbonate chloride exchanger because one negative ion is going or secreted into the interstitial fluid site. So one should be enter inside the parietal cells. Is it clear to everyone? Now, what will happen to this hydrogen? Now, this hydrogen will also secreted towards the lumen site in the exchange of ion, that is potassium, through a channel that is known as hydrogen potassium ATPase. The name of the channel is very, very important. You have to remember this, right? Hydrogen potassium ATPase. Now, this chloride, this will travel and this will also secreted towards the lumen Please tell me if the screen is visible to you, everyone. Slides are visible. Please confirm.
No, ma'am.
Wait. Is it now visible?
Yes, ma'am.
Okay. So as we have seen that CO2 is derived from the metabolic activity of the parietal cells also, and some CO2 also we get from the diffusion of blood vessels also. So it combines with water to form the very important substance that is known as carbonic acid in the presence of an enzyme known as carbonic anhydrase. So this enzyme is present in high concentration in parietal cells. So carbonic acid is much unstable compound, and basically it immediate split into bicarbonate and in the H plus. Right now, the H plus is pumped actively in the exchange of potassium with the help of an channel known as hydrogen potassium ATPase. So why ATPase? Because it hydrolyzes 1 ATP in this activity. Now the simultaneously the chloride ion is also pumped towards this lumen side or canicular side, and now the hydrogen ion and chloride ion will combine and together they will form HCl. So I think now the concept of mechanism of HCl secretion is very well clear to the students. Another thing, we have the regulation of acid secretion. So basically, the acid secretion, for the regulation, three things are very important. First is acetylcholine, second thing is histamine, and the third is gastrin. So already we have seen the gastrin are secreted by the cells known as G cells. Histamine is secreted by enterochromaffin-like cells, as we have seen. Then acetylcholine is produced by the cholinergic nerve terminals. So now these three things or these three hormones are basically responsible for binding over the receptors. Now what are those receptors? The acetylcholine will bind over the M3, or another name of M3 is muscarinic. Right? So histamine will act over the hydrogen receptor, and gastrin will bind over the CCKB receptors, and together they are responsible for increasing the production of H plus ion so that the formation of HCl can be take place. So this is all about the regulation of acid secretion. Now, as we have seen, the factor affecting acid secretion, the first we have gastrin secreted by G cells, then histamine secreted by enterochromaffin-like cells, and the vagal stimulation by the cholinergic nerve endings. Now, as we can see for the cephalic phase, so as we have discussed in the saliva also, so basically the cephalic phase is the phase in which there will be start secretion whenever the food enters inside the mouth, right? In the conditioned reflex or in the unconditioned reflex, whenever the food is present in the thought only, thought through the thought, smell, or by seeing. So what will happen whenever in unconditioned reflex, basically here we're talking about whenever we saw the food, we smell the food, or we saw the food, or whenever the food is present inside the mouth, means during taste in the conditioned reflex of cephalic phase, there will be stimulation of vagus nerve, and through vagus nerve what will happen, through enteric nerve plexus there will be stimulation of these cells to produce different hormones so that production of HCl can be increased, as we have seen in the regulation part also, right? Now the gastric phase acid. So basically through this cephalic phase, what we get to know that basically what happened in cephalic phase, we have two different type of reflexes. The first we have conditioned reflex, and the second we have unconditioned reflex, right? In conditioned reflex means the food is present in the mouth, and in unconditioned the food is present in the thought only, right? So I have a very important question for you, and have 30 seconds to answer. Okay, so this is the question. Clinical case scenario, patient is present with gastric pain and discomfort meal. Upon further investigation, it is suspected that symptom may be related to disturbance in gastric secretion. So basically, they are asking during the cephalic phase, which of the following stimuli is most likely for the production of gastric secretion in the patient. So you have 30 seconds to answer. The option is presence of chyme in the stomach, side smell or thought, distension of the stomach, release of gastrin from the duodenum. Now you need to remember a very important thing, that they are asking during cephalic phase only. And this clue has the answer. You can answer in the chat box also. Option A, B, C, D. Vidya, do you know the answer? Okay, Neelajit. Am I audible to you all? Yes, the answer is B. Sight, smell, or thought of food. Yes, correct. Answer is B. Okay, now the phases of gastric secretion, another very important thing. So basically, in gastric secretion, we have three phases. The first we have cephalic phase, the second we have gastric phase, and the third we have intestinal phase. So as the name suggests, cephalic is head, right? So basically, this phase starts whenever the food present in the throat or it is present in the mouth. Okay, so for this, we have two reflexes. The first we have conditioned reflex. It is associated with sight, smell, and thought of food and hearing about food also. Then in unconditioned reflex, whenever the food is present inside the mouth. Now, both the reflexes basically will stimulate the 10th cranial nerve, that is vagus nerve. As we know, the main function of the vagus nerve to bind upon M3 receptors and to increase the HCl or to increase the production of gastric juice, right? Now, the another thing we have gastric phase. So as again, the name suggests whenever the food is present inside the stomach, right? So whenever the food is present, the bolus is present in stomach. Again, vagus nerve, some local nerve and gastrin will be there. Both will stimulate the production of gastric juice, right? Then in intestinal phase, whenever the food is present inside the intestine, the chyme is present inside intestine. Now there will be stimulation or production of gastrin will increase, which will stimulate secretion of gastric juice. Another thing, some hormones are also present for the inhibition of this gastric juice, right? And in which we have secretin, CCK, somatostatin, GIP, and BIP. All these hormones are responsible for inhibition of gastric juice, right? So in intestinal phase, we have gastrin for the stimulation or for increasing the secretion of gastric juice, and for the inhibition, we have these hormones, right? So again, I'm briefing you about the phases of gastric secretion. So basically, the cephalic phase of gastric secretion is the secretion of gastric juice by the stimuli arising from the head region, right? Means cephalic means head, such as through sight, smell, or thought of food, or the presence of food inside the mouth. during this phase, gastric secretion occurs even without the presence of food in mouth also, in stomach also, sorry. So that's why we call that sometimes acidity also occurs inside the stomach without food. This is the reason, right? So gastric juice secreted during this phase is known as appetite juice. Now the quantity of the juice is less, but it is rich in enzyme and it's HCl. So basically, during this phase, there will be 30% of total amount of gastric juice will be secreted. Clear? Then another thing we have the gastric phase. So basically the gastric phase of gastric secretion is the secretion of gastric juice when food enters inside the stomach, as we have already discussed. Now the gastric juice secreted during this phase is rich in pepsinogen and HCl. And if we talk about the total amount, so basically here 60% of the total amount of gastric juice is secreted and it is controlled by both nervous and the hormonal mechanism. So for the hormonal, as we know, this is controlled by the G cells, right? G cells which produce gastrin. So gastrin is a gastrointestinal hormone secreted by the G cells which are present in the, as we have discussed, in the pyruvate gland of the stomach. Small amount of gastrin is also secreted in mucus of upper small intestine also. Then we have some mesenteric plexus and vagal reflex also, through which there will be stimulation or there will be increase in the production of or secretion of gastric juice. Then the last we have intestinal phase. So basically the intestinal phase is the phase in which there will be secretion of gastric juice when chyme enter inside the intestine, right? And in the intestinal phase of gastric secretion is regulated by nervous and hormonal, okay? And if we talk about the percentage, so basically here we have only 10% of total gastric juice is secreted. So if I ask you in which phase there will be highest secretion of gastric juice will occur, so the answer will be gastric phase. Clear? So basically, in the intestinal phase, it has two stages, right? The first stage in which there will be stimulation of G cells and gastrin will produce to increase the production of gastric juice. And in the later stage, there will be production of these hormones which inhibit the production of gastric juice, right? So with this, we have covered the stomach, the whole stomach. Then we have some motilities. So basically for the motility, first we study for motility, first we study the migrating motor complex. So basically what is this MMC? This is the periodic intense electrical activity that is seen in the empty stomach or small intestine that lasts for 3 to 6 minutes and spread from stomach to ileum. So basically that reflex will take all the food from stomach and pass it into last part of the small intestine, and it is cyclical and repeated in every 90 minutes. Now it has three phases. The first is the phase 1, the second is phase 2, and the third is phase 3. So in the diagram also we can see in the first phase slow waves are there, right? And in the second phase, irregular are there. The intervals are very irregular, but it has spikes, right? So spike means the activities are little bit intense. And in the third phase, as we can see, the waves are not as such irregular, but the spikes are so many. So basically in this phase, in third phase, the activity will be very intense of MMC. So, migrating motility complex is the housekeeping function that sweeps undigested residue toward colon to maintain low bacterial count in upper intestine. It is the most coordinated rapid peristalsis which occur between meals. The characteristics are period of intense peristaltic contraction takes 90 minutes to go from stomach to colon and mediated by motilin, which is a protein secreted by MO cells and anteric nervous system. Okay, so I have some poll questions for the students. You have 30 seconds to answer. In human body, the digestion of protein begins in which part of the following organ? Liver, mouth, small intestine, and stomach. Please answer. Poll questions are visible. Yes, the answer is stomach. All are launch site. Okay, next thing we have gastric emptying and mixing. So both are the very important factors. So basically gastric emptying means whenever the food get digested inside the stomach and it will travel to the small intestine, and mixing means basically a wave that is present for the mixing of food with the juices that is present inside the stomach. So gastric emptying is the peristaltic contraction originate in the upper fundus and sweeps down towards the parotid sphincter. The contraction become more vigorous as it reaches the thick muscle antrum. The strong antral peristaltic contraction propels the chyme forward. A small portion of chyme is pushed through the partially open sphincter into the duodenum. The stronger the antral contraction, the more chyme is impinged with each contractile wave. Then, as we have discussed, mixing means mixing of food with the juices, right? So where the peristaltic contraction reaches the parotid sphincter, the sphincter is tightly closed and no further impinging take place. Then the chyme that was being propelled forwards, this hits the closed sphincter, it is loose back into the antrum. Mixing of chyme is accomplished as chyme is propelled towards the antos back into the antrum with each peristaltic contraction. So the regulation of gastric emptying basically associated gastric food volume increase, food volume in the stomach increase, gastric emptying. Then fluidity also increase, fluidity allows more rapid emptying. Then hormone gastrin has a very important role. Basically, it stimulates motor function in the body of the stomach and hence the peristaltic contraction. Now, the secretion of gastrin stimulated by stretching of stomach and with the presence of certain types of food that is also present in stomach, especially meat products. And then these things already we have discussed. Okay, now coming to the applied physiology. So we have a very important condition that is known as gastritis. So basically it is the inflammation of gastric mucosa that is known as gastritis, and it could be acute and chronic. It results in the atrophy of gastric mucosa with loss of chief cells and parietal cells, both will be loose in this case, so that the secretion of gastric juice ultimately decreases. Another thing we have peptic ulcers. Peptic ulcers means erosion of the surface of any organ due to shedding and slowing of the inflamed necrotic tissue. Necrotic tissue, basically the dead tissues that lines the organ. If peptic ulcer is found in stomach, so it is known as gastric ulcer, and if it is found in duodenum, so it is known as duodenal ulcer. Increased peptic activity due to excessive secretion of pepsin in gastric juice, hyperacidity of gastric juice, reduced alkalinity of duodenal content, decreased mucin content in gastric juice, or decreased protective activity in stomach or duodenum. Then we have constant physical or emotional stress fueled with excess spice or smoking also causes this type of ulcers. Then chronic inflammation is due to presence of H. pylori. Then most common feature of peptic ulcer is severe burning. In gastric pain, in gastric ulcer, pain occurs while eating or drinking. In duodenal ulcer, pain is felt 1 to 2 hours after food intake, and especially during night. So symptoms we have the vomiting, heartburn, nausea, loss of appetite, and loss of weight. So this is all about today's topic. Hope this class is very useful to the students. So thank you so much.